Welcome to the deep dive. We're diving into some really cutting-edge cancer immunotherapy research today.
Really exciting stuff.
Yeah, we're going to be breaking down a 2024 Nature article. And,
Right. Oh, nature.
when it's Nature,
That's the big leagues.
it's a big deal.
Yeah.
So this one is called the Type 2 cytokine FcIL4 Revitalizes exhausted CD8 plus T cells against cancer.
By Feng et al.
That's right, Feng et al. And for our listener who's a new PhD student in immunology, we're going to maybe go a little bit deeper on some of the technical details.
Absolutely. We'll throw in some lab tips and tricks along the way.
Perfect. So this paper is all about T-cell exhaustion, which is a major challenge in cancer immunotherapy. Can you give us a quick overview of what that is?
Sure. So imagine your immune system is kind of like an army. And your CD8 plus T cells are like the elite special forces. They're trained to seek and destroy invaders like cancer cells. But fighting a prolonged battle takes its toll, and these cells can become exhausted.
So it's like burnout, but for immune cells. They lose their punch even when we have treatments like ICB or ACT that are trying to rev them up.
Exactly. And those therapies are amazing. But T-cell exhaustion is a major thing that limits their effectiveness sometimes.
Right. So that's where this FcIL4 comes in. And this is where things get really interesting.
This is where it gets cool.
Instead of just focusing on type 1 immunity, which is what we traditionally target in cancer treatment, FcIL4 is tapping into type 2 immunity.
Yeah, it's a total paradigm shift.
So what is that?
So FcIL4 is a fusion protein, and it combines interleukin-4, or IL-4, which is a type 2 cytokine, with an antibody fragment.
Okay, let's pause there for a second. For our listener who's maybe new to this, what exactly is a cytokine, and how do these things actually work?
Okay, so cytokines, you can think of them as the messengers of the immune system. They're signaling molecules that tell cells what to do. It's kind of like instructions written in a specific code. And in the lab, we can use them to stimulate immune cells or block their activity, depending on what we're studying. And IL-4 is a cytokine that's involved in type 2 immune responses.
Okay, so this fusion protein is essentially delivering IL-4 directly to specific T cells. And in this case, it's a subset called CD8 plus TTE cells. So what makes these TTE cells so special?
So these cells are terminally exhausted, so they've hit a wall in terms of their functionality. But the catch is they still have this potential to kill cancer cells.
Oh, wow.
The challenge is that their numbers are really low and they struggle to survive in the harsh tumor environment.
So they're kind of like...
They're there, but they're not really doing much.
They're there, but they need some help.
Exactly.
So FcIL4 is kind of coming in and revitalizing them, like turning them into these super-powered cancer-killing machines.
That's exactly what the research suggests. So in various mouse models, they found that treatment with FcIL4 led to a big increase in these CD8 plus TTE cells within the tumors.
Okay.
And it also boosted their ability to produce granzyme B. And granzyme B is a molecule these cells use to destroy cancer cells.
That's awesome. So we're seeing some serious tumor fighting potential here. But how is it actually doing that? How is FcIL4 actually making these T cells stronger? Like, what's going on at the cellular level?
Well, this is where it gets really interesting. So the researchers found that FcIL4 boosts the metabolic activity of these cells. Specifically, it promotes something called glycolysis and increases the level of NAD+.
Okay, so let's break that down a little bit. Glycolysis first. That's how cells break down glucose for energy, right?
Exactly. It's like a fundamental metabolic pathway.
Right.
And it's essential for cells to function properly. I mean, you need energy to do anything.
It's like fueling up before a big game.
Exactly. So, for these T cells to effectively kill cancer cells, they need a lot of energy.
They got to be ready to go.
Yeah.
Okay, that makes sense. But what about NAD plus air? That one sounds a bit more complicated.
Yeah, so NAD plus stands for nicotinamide adenine dinucleotide.
Catchy.
Yeah, it's a mouthful. But it's basically a really crucial coenzyme that's involved in lots of cellular processes, including energy production and DNA repair.
So it's like a vital ingredient that keeps these T cells healthy and running smoothly.
Exactly. And FcIL4 increases these NAD plus levels, which helps boost the overall function of these exhausted T cells.
Okay, so we've got glycolysis and NAD plus working together to kind of supercharge these T cells. But is there like a specific molecule that's driving this whole process?
That's a great question, and this is where the research gets even more exciting. So they identified a key enzyme called LDHA, which stands for lactate dehydrogenase A, and that plays a crucial role in this whole metabolic boost.
Now, LDHA, that sounds familiar. I feel like we've talked about that in a previous deep dive.
It comes up a lot.
Yeah, it's a pretty important player in cellular metabolism.
Yeah, it is.
So in this study, they found that FcIL4 specifically increases the activity of LDHA in these CD8 plus T E cells.
Right.
So LDHA is like the engine behind this metabolic boost.
Exactly. The increased LDHA activity then drives both glycolysis, giving those T cells more energy, and NAD plus production, so ensuring they have the resources to function properly.
Okay, so LDHA is like the star of the show here. But how did the researchers actually figure that out? Like, I imagine there was some pretty sophisticated lab work involved.
Oh, for sure. So to confirm LDHA's role, they used a specific LDHA inhibitor. It's called FX11.
Okay.
And they even knocked out LDHA completely in some of their experiments.
Now, for our listener, can you explain what it means to knock out a gene?
Yeah, so gene knockout is basically a technique where we delete or inactivate a specific gene in an organism. So in this case, they were working with mice. And this lets us study the function of that gene by seeing what happens when it's missing.
So by knocking out LDHA, they were able to see what happens to those T cells when LDHA is no longer functioning.
Exactly. And what they found was really interesting. When LDHA was inhibited or knocked out, the beneficial effects of FcIL4 were significantly diminished.
So without LDHA, those T cells weren't getting the same metabolic boost. And their ability to fight cancer was weakened.
Exactly, which really highlights how critical this LDHA is in this whole process.
All right, so for our listener, this knockout experiment is a really cool example of how we can use genetic manipulations in the lab to understand the function of specific genes and proteins.
Yeah, for sure.
It's amazing how we can literally manipulate genes to uncover these secrets of cellular processes.
It's really remarkable, and this is just the beginning.
I know there's so much more to talk about.
Yeah, this is just the start of our journey into this paper and all the cool lab techniques that they use.
Absolutely.
Yeah.
We've got a lot more to unpack.
Yeah.
So stay tuned. We'll be back soon with part 2 of this deep dive.
Sounds good.
Welcome back to the deep dive. We're continuing our exploration of FcIL4 and all its potential for cancer immunotherapy.
Yeah, it's really amazing research, and we're just getting started with the lab techniques that they use.
Right. Last time we left off talking about LDHA and how it's involved in boosting the metabolism of those exhausted T cells. But I remember the paper also mentioned something called the NAD plus salvage pathway.
Oh, yeah.
What is that all about?
So the NAD plus salvage pathway is basically a recycling system for NAD plus within the cell.
Okay.
So instead of having to make new NAD plus from scratch all the time, the cell can actually reuse and replenish its existing supply.
Oh, so it's efficient.
Yeah, it's super efficient.
Gotcha. So how do you actually study this pathway in the lab? What kind of techniques did the researchers use here?
So they did something pretty clever. They supplemented the CD8 plus TTE cells with a molecule called nicotinamide riboside, which is actually a form of vitamin B3.
Nicotinamide riboside. That sounds familiar. How does that tie into NAD plus go?
So nicotinamide riboside is a precursor to NAD+. So you can think of it like a building block that the cell can use to create more NAD+.
Okay.
It's like providing the raw materials for a factory.
Gotcha.
And they wanted to see if this direct boost of NAD+ would have a similar effect to FcIL4.
Oh, interesting. So they're kind of like bypassing that whole LDHA glycolysis route and just going straight to the source.
Exactly.
So what did they find?
So they found that just like with FcIL4, they saw an increase in glycolysis, indicating that those T cells were getting a metabolic boost even without FcIL4 directly stimulating LDHA.
So they're able to kind of enhance metabolism by just directly increasing NAD plus levels.
Exactly.
Interesting. Did that actually translate into improved T cell function, though? Like, did those cells become better at fighting cancer?
Yeah, that's the key question, right? And the answer is yes.
Okay, great.
They saw an increase in T cell expansion, so those cells were dividing more, and they also saw enhanced cytolytic activity, meaning they were better at killing cancer cells.
That's incredible. So even without FcIL4, directly involved just boosting NAD plus levels, had this positive effect on T cell function.
Right.
That's really cool. So I guess this opens up a whole new avenue for potential therapies, right?
Absolutely. And that's one of the most exciting things about this research. It's not just about FcIL4 itself. It's about understanding these underlying mechanisms and pathways that we can target to enhance the immune response against cancer.
It's like we're uncovering the secrets to how to make these exhausted T cells wake up and fight again.
Exactly.
That's awesome. So I'm curious, did the researchers just add the nicotinamide riboside to the cells in a dish? Or did they use any other techniques to study this in vivo?
So they actually did both. So in addition to in vitro experiments, they also tested nicotinamide riboside in mouse models. And they found that supplementing the mice with nicotinamide riboside enhanced the anti-tumor activity of T cells,
Okay.
especially when it was combined with other immunotherapies like ICB.
Wow. So this NAD plus boost isn't just theoretical, it's actually showing these promising results in living organisms.
Right.
That's really cool.
Hmm.
But I think it's important to acknowledge that, every scientific breakthrough comes with its limitations.
Yeah.
What were some of the caveats or areas for further research that the authors highlighted?
So one of the big things to remember is that this research was primarily done in mouse models.
Right. Mouse models are great, but they don't always perfectly translate to humans.
Exactly. So while these results are super promising, it's important to be cautious. Yeah. And wait for further research to confirm whether these same effects will hold true in humans.
That makes sense.
Yeah.
Were there any other limitations that they mentioned?
So another thing they emphasized was the need to better understand the long-term durability of the immune response.
Okay, so how long do these effects actually last?
Right, because the study showed some pretty amazing results. Some of the mice even achieved durable cures, but we need to know, how long do those benefits last, both in terms of controlling tumor growth and establishing that long-lasting immune memory.
Right, so it's not just about winning the initial battle. It's about making sure the immune system is prepared for any future encounters.
Exactly, and that's an area where the researchers acknowledged that more research is needed.
Okay. Anything else?
They also highlighted the importance of understanding the potential interplay between FcIL4 and other parts of the immune system.
Right, because the immune system is not just like one thing, it's this whole complex network.
Exactly. It's like an orchestra with all these different cells and molecules playing specific roles.
It's a beautiful orchestra.
It is. And while this study focused mainly on CD8 plus T cells, we need to understand how FcIL4 might be impacting other immune cells, both within the tumor microenvironment and systemically.
So it's like we've identified one key player in this orchestra.
Right.
But we need to understand how it fits into the larger ensemble.
Exactly. And make sure we're not, disrupting the balance of the whole system while we're trying to enhance its ability to fight cancer.
It's all about finding that balance, that harmony.
Exactly.
Okay, so we've talked about limitations and areas for future research. But I want to touch on one more thing before we wrap up this part of our deep dive. The paper mentioned that FcIL4 seems to be effective even in the absence of progenitor-exhausted T cells. Can you explain what that means and why it's significant?
Yeah, so progenitor-exhausted T cells are kind of like the main targets of existing immunotherapies like ICB.
Okay.
They're the ones that still have some fight left in them. And those are the ones that we try to reinvigorate with things like checkpoint inhibitors.
Right, they're like the reserves.
Exactly. But what's really cool about this study is that FcIL4 was able to enhance anti-tumor immunity even when those progenitor cells were depleted.
Oh, wow. So it's like it's bypassing the reserves and going straight to the front lines?
Exactly.
And revitalizing those terminally exhausted cells that were previously thought to be too far gone.
Exactly. And this has some pretty big implications.
Yeah.
It suggests that FcIL4 might be effective in patients who haven't responded well to traditional immunotherapies because their progenitor-exhausted T cells are just not functional anymore.
That's incredible. It's like we've discovered a way to rally those veteran soldiers who we thought were out of the fight.
Yeah, and it highlights how amazing the immune system is. It's so plastic and adaptable that even when one pathway is blocked, there are often alternative routes that we can explore to enhance its ability to fight disease.
This is really mind-blowing stuff.
It is.
But I think we've covered a lot of ground for now. What do you say we take a break and come back for part 3 where we can delve into the ethical considerations and future directions of this research?
Yeah, that sounds great.
Welcome back to the deep dive. We've been on quite a journey exploring FcIL4 and its potential to really shake things up in cancer treatment.
It's amazing stuff.
It really is. From those exhausted T cells to the whole NAD plus thing and that salvage pathway, we've covered a lot of ground.
A lot of really cool lab techniques, too.
Yeah, for sure.
Yeah.
So now for this final part, let's zoom out a little bit and talk about the bigger picture.
Okay, sounds good.
What does this research actually mean for the future of cancer treatment? Like, where do we go from here?
Well, I think this research is like a beacon. It's really illuminating these new paths in cancer immunotherapy.
Ooh, I like that analogy.
Yeah.
But before we get too carried away with the excitement, I think it's important to acknowledge, the ethical considerations around this kind of research. We're talking about manipulating the immune system, which is powerful stuff. And we need to be really mindful of those implications.
Absolutely. I think one of the most important things to consider is access and equity.
Mm-hmm.
If FCIL4 turns out to be as effective in humans as it has been in these mouse models, we need to make sure that this treatment is available to everyone who could benefit from it,
Right.
regardless of, their background or where they live.
It can't just be a treatment for the privileged few. We need to make sure it's accessible to everyone.
Exactly.
What about other ethical concerns? Are there any potential risks or unintended consequences that we should be thinking about?
Well, whenever we're developing new therapies, especially ones that involve manipulating the immune system, we always need to be on the lookout for off-target effects. Even though FCIL4 seems to have a very specific effect on those CD8 plus TTE cells, there's always the possibility that it could impact other cells or processes in ways that we haven't even anticipated.
So it's like we're playing with a really powerful tool, and we need to make sure we're using it carefully and responsibly.
Exactly. Thorough testing is absolutely crucial, and we need to be really cautious as we move into human trials, carefully monitoring patients for any signs of, bad reactions.
Yeah, safety first.
Absolutely. And informed consent is really important too. Patients need to fully understand the risks and benefits before they decide to participate in a trial.
Definitely, transparency and open communication are so important. But let's switch gears a bit and talk about the future. This research is clearly groundbreaking, and it opens up all these exciting possibilities for cancer treatment. Mm-hmm. What are you most excited about? What are some of the things you're really looking forward to?
Well, one area that I think holds a lot of promise is personalized immunotherapy.
Yeah, I've heard a lot about that. What is that exactly?
So as we learn more and more about the unique characteristics of each patient's immune system and their tumor, we can actually start to tailor therapies to their specific needs.
So instead of a one-size-fits-all approach, it's more like creating a custom treatment plan.
Exactly, like a custom-made suit of armor for their immune system.
I love that. So are we already seeing this happening?
Yeah, we are actually. We're seeing some progress in this area with things like CAR T-cell therapy, where T-cells are engineered to target very specific cancer antigens. But with advancements in gene editing technologies like CRISPR, the possibilities for personalized immunotherapy are just exploding.
That's amazing.
Yeah.
It's mind-blowing to think about what we might be able to do in the future. What else are you keeping an eye on?
Another area I'm really excited about is combining immunotherapy with other types of treatment.
Okay, so what does that look like?
So imagine combining, the precision of targeted therapies with the power of immunotherapy, or using chemo to weaken the tumor and then unleashing the immune system to finish it off.
So it's like we're building this arsenal of weapons, each with its own unique strengths, and we're learning how to use them strategically.
That's a great way to put it. And that's really what's happening in the field right now. We're not thinking about these treatments as separate things anymore. It's all about combining them into a comprehensive strategy.
Right, it's like this whole new way of thinking about cancer treatment.
Exactly.
And the research on FCIL4 is a perfect example of that.
Yeah.
It's not about replacing existing therapies. It's about figuring out how to make them work even better.
Right. It's about understanding this really complex language of the immune system and learning how to speak it.
Like we're becoming immune system whisperers.
I love that analogy. And I think as we continue to learn more about this incredible system, we're going to discover even more innovative and effective therapies, and we're going to get closer and closer to a future where cancer is no longer this feared and deadly disease.
That's a beautiful thought. This has been such an incredible deep dive. I'm feeling so energized and hopeful about the future of cancer treatment. Thank you so much for sharing your expertise with us today.
It's been my pleasure. It's always exciting to talk about this kind of groundbreaking research, and I'm so glad we could share it with our listeners.
And to our listener who is just starting their journey as a PhD student in immunology, we hope this deep dive has given you a glimpse into this amazing world of cancer immunotherapy and all the amazing tools we're developing to fight this disease.
Remember, the future of medicine is being shaped by passionate and curious minds like yours. So keep asking those questions, keep exploring, and never stop pushing the boundaries of science.
And to all our listeners, thank you so much for joining us on the deep dive. Be sure to check out the show notes for a link to the full research article. And don't forget to subscribe for more fascinating journeys into the world of science and technology.
Until next time, stay curious.